we're interested in the evolution of phenotypic diversity. We want to know how so many different forms have evolved. And from a phylogenetic perspective, of course, remembering that all of these extant species relate back through time through a single giant tree, their differences relate to all the mutations that have accumulated towards their path to the present. So we have this great challenge of figuring out what happened along their path to the present and which of the genetic changes along their path contributed to their phenotypic differences. And so that's the grand challenge we're here to talk about. So I wanted to open by reminding you of some of the ways that we have classically addressed this problem, and then dividing it into sort of three major categories: population genetic approaches, molecular genetic approaches, and comparative or phylogenetic approaches. So probably most of you are very familiar with QTL mapping, where you take two lines that differ in some trait of interest, and you cross them to get populations where the genes and the trait is segregating in that population, and you can look for associations. And so this is one of the most powerful approaches, and it's genome-wide. It doesn't require that you have candidate genes, but it does require that you have species that you can cross. And so I'll return back to what the consequences of that might be, but that's been a very common approach. Another population genetic approach is genome-wide association mapping. This is often used in cases where you can't do controlled crosses, so for example with humans, and you have to rely on naturally segregating variation to look for those associations between genes and phenotypes of interest. And there's a large literature about the challenges of GWAS, but there are often genes that stand out in these, such as the eye color and glaucoma genes. So one challenge both with QTL mapping and with GWAS is that often they get you to a part of the genome, but they don't get you to a gene or a genetic change. Some of the instances in which we've been most successful in actually getting to the genes and the genetic changes are cases where we know a lot about the actual development of those traits. So this is just an example that I happen to know. This is the flower development pathway that was worked on in Arabidopsis and subsequently in a number of other model systems. And so when we have this kind of pathway, we can often relate it to QTL maps and ask whether some of the genes in that QTL interval correspond to genes in these pathways. So what's important to remember is that these pathways tell us how a structure is made, but they don't necessarily tell us how the structure evolves, how is it changing. So we have to take a comparative approach to address that question. So for example, we might compare two species that differ in trait. Here's two species that differ in having or not having petals, and then we can look at this pathway and ask what's different. And in this case, the species that has no petals has a deletion of one of the floral development genes. So what we really like to do is expand across entire phylogenies. And when we have these cases of well-known pathways, we can look at candidate genes and consider how their expression corresponds to phenotype. This is a talk from my lab that was given earlier today, where we've looked at a couple of genes involved in floral pigmentation, and we see that in lineages that lack floral pigmentation, it's repeatedly downregulated. So sort of emphasizing both the role of expression variation and these particular loci in that type of phenotypic shift. But obviously, we would all love to move beyond candidate genes, because building pathways like the one I showed you requires decades of detailed work in model systems, and we simply are not going to have time to develop that kind of detailed understanding of too many taxa. And so really my hope and what I want to ponder collectively today is if there are ways that we can take some of the strengths of population genetic approaches, of molecular genetic approaches, and put them into a comparative framework. Not just allow to have a comparison. I do a QTL map, I find some region, and then there's this candidate gene in the region, but more directly integrate them into a complete framework. And so those are some of the approaches we're going to be talking about today. And I'd like to spend a few minutes advocating why this is a useful, if challenging, endeavor. One is that there's lots of traits that we want to learn about how they evolved that happened too long ago for us to cross the taxa. We're no longer going to be able to cross anything that does and doesn't have wings. We cannot QTL map wings. So we're really limited in what kinds of traits we can apply those population genetic approaches to. The other thing is I think that something like the QTL map tells us what are the range of genetic changes between these two species, but it doesn't tell us about the assembly of those changes. Which came first, which came last? What was the ancestral state? What's the derived state? Those are really stories that phylogeneticists like to tell, but I think all evolutionary biologists want to understand these evolutionary trajectories. And by putting it into a tree, we can begin to break down the steps towards some of these complex phenotypic shifts. And finally, there's been a lot of attention on molecular convergence. How often are the same traits evolved by the same underlying molecular changes? But I think we also want to know about the non-convergence. How often do lineages take different routes to get to the same phenotype? And we really need to work at a macroevolutionary scale to arrive at a quantitative sense for that. The last pitch I would make for these sort of approaches is that the tree of life is really big, and it's full of all of these really interesting and unique phenotypes. And working piecemeal, shallow work in QTL mapping, or deeper work where we look at comparative expression, but we're assuming the pathway is the same. Those kind of approaches are really going to limit how far we can walk across the tree. If we really want to walk out to diverse groups, we need to have methods that don't necessarily rely on a lot of genetic resources or on knowing a lot about the genetic basis of particular traits. And so with that, I'm going to turn it over to Casey.
All right, so as these molecular genetic approaches are applied at scale to genome level data, they take on this new name, functional genomics. It's one of those things that can mean slightly different things to different people, but it certainly includes a lot of methods that folks in this room are familiar with, like RNA-seq, ChIP-seq, and other approaches that could be applied across many genes. And with the power to collect these functional genomic data, especially in non-model organisms, we're seeing a real explosion of comparative functional genomics as a new field. And these analyses integrate functional genomic data across species to investigate the evolution of genome function and to do exactly what Stacey was talking about, to associate genes and phenotypes. So the enthusiasm for this approach is apparent in a growing number of comparative functional genomics papers. This is just a smattering of them. I haven't plotted it out, but they're growing exponentially. This is becoming a very popular approach. But there's a big potential problem with the way comparative functional genomics is being deployed right now. And that's that most of these studies rely on pairwise comparisons between attributes of genes across species and across homologs within gene trees. So folks in this room certainly know why this can be problematic to make pairwise comparisons between species. So these approaches don't take into account evolutionary relationships, and they're therefore not independent. And so each branch is included in multiple comparisons. So you're getting the exact same evolutionary changes cropping up in multiple pairwise comparisons. And also these pairwise comparisons are a very blunt tool because they're looking at very distantly related genes often, and they can't really pinpoint where on the tree the changes are happening. Fortunately, people have been thinking about this a lot, but in other contexts, in the context of comparative morphology, comparative physiology, but we have this really rich set of phylogenetic comparative methods, including independent contrast, of course, that do take these relationships into account. They're just not being used in this new field, and we've repeatedly seen this, where new comparative fields start out with pairwise comparisons and then realize there's problems, and then move on to phylogenetic comparative methods. So we need to move on to phylogenetic comparative methods as soon as possible with these comparative functional genomics analyses. So this begs the question, does it matter? So maybe there's something about comparative functional genomics where the biases that can impact pairwise comparisons aren't a big deal. So that's a perfectly valid, proposition coming from someone who is doing functional genomics but isn't yet familiar with comparative methods. So we recently did some analyses where we took a look at 2 recently published comparative functional genomics studies. And the take-home, so the spoiler, is that in both cases we found fundamental flaws with the pairwise methods that had very big impacts on the conclusions of those papers. So I'm going to talk today about the reanalyses we did for this top paper here. And this is available in the preprint. So this is work that was done by me, Felipe Zapata, who's here, and then some other folks, some other collaborators and folks in our labs. So the source code for all these analyses is available here, and I put a little link here to just a sheet that has notes with links to all the papers we're talking about here and code repositories. So this paper, tissue specificity of gene expression was a test of the ortholog conjecture. So 2 genes are orthologs if they diverge due to a speciation event, and 2 genes are paralogs if they diverge due to a gene duplication event. So orthologs and paralogs are just specific instances of homology. So paralogs and orthologs are all homologs. This is a specific type of homology that we talk about. And orthology and paralogy have become absolutely central to the way we discuss the diversity and evolution of gene function. You will not find a comparative genomics paper that isn't talking a lot about paralogy and orthology. So that would indicate that there's this expectation that these terms explain a lot about the diversity of function that we see in genes. So understanding how the evolution of gene functions differs between orthologs and paralogs is really fundamental to understanding the association between genes and phenotypes in the way these problems are presented today. So the ortholog conjecture is the hypothesis that orthologs are more similar to each other than paralogs are. So the expectation is that if you have a set of homologs and you pick one gene in there that you know a lot about, the orthologs to that gene are going to be more similar functionally than the paralogs to that gene are within that same tree. So that's a pattern. That's a description of a pattern. It's also articulated sometimes in terms of a process. So gene evolution is more rapid after duplication than speciation. All right? And there's some really interesting specific models why we might think this is the case, like degeneration and complementation and things like that. And those models have been shown to play out in specific cases. Really, the ortholog conjecture is a generalization of those models, saying, in general, we should expect those processes to have a big impact, so that orthologs should always be expected to be more similar than paralogs. The ortholog conjecture has received mixed support. There's been a really nice series of analyses going back and forth, and sometimes people are talking about in the context of expression, sometimes about chemistry, sometimes sequence. So the paper that I'm talking about here found strong support for the ortholog conjecture with regard to gene expression. So they examined tissue specificity of gene expression with this metric tau, where tau is 1 if the expression is entirely in one tissue type, and it's 0 if it's uniform. So it doesn't actually preserve information about which organ or tissue that the expression is in, but it's telling you about the specificity. And they looked across multiple public data sets of mammal organ expression, and they used pairwise comparisons across genes and species. This is the really important figure here. On the x-axis, we have divergence time as indicated by specific clades. So the nodes corresponding to those clades. Divergence time is the age of those nodes. And then we're showing the correlation of tau in sets of genes here. And so what we can see here is that there's much higher correlation in tau between orthologs that diverged at the same depth in the tree versus the paralogs that diverged at the same depth in the tree. So given the depth in the tree, there's more correlation. So they took this to indicate that if you know what one ortholog is doing, you're much more likely to know what another ortholog is doing rather than knowing what a paralog is doing. One thing to note here is that they didn't show the actual divergence time of every duplication. They lumped the duplications together into a speciation node that was close by on the tree. So the first thing we did when we got their data set was that we made sure we could reproduce their original result, and we could. They did a great job putting all the code and data out there. It just took a few hours to actually regenerate their plots with some of our own code. So you can see we have the genes arising from speciation, so the orthologs here have higher correlation coefficients than the duplicates do. So we can get that result. And then we reanalyzed their data with phylogenetic independent contrast, so the very well-established comparative method. And what we did here is a little bit different than how contrasts are usually used. Usually you're looking at the association of multiple characters on the tips of the tree. Here we bin those contrasts in a single character, tau, according to whether the node that contrast spanned was a duplication or a speciation node. And the larger the contrast, the more change per unit branch length there's been since that node. So the specific expectation, the prediction of the ortholog conjecture, this particular hypothesis, is that in the presence of this increased rate of change following duplications, you would see an excess of large contrast for duplicates if you look at the density plots here. So you can see there's proportionally more of the smaller contrast for speciation events and proportionally more of the larger contrast for duplication events. And then if the alternative hypothesis, the null, would be that those distributions are the same, or that those contrasts are drawn from the same distribution. So to make it a little bit clearer, sometimes it's hard to see the distances between these lines. I actually like looking at the difference in the density distributions. So on the right here, you can see that there is just this excess of duplication events. So there's this, an excess of large contrast for duplication events. So there's that large bulge behind the line. So that's a prediction of what you see, and you just sort of see this wiggly flat line under the null expectation. So what do the data look like? So when we reanalyze that exact same data set with these phylogenetic methods, we actually get a very different pattern. We actually see an excess of larger contrast following speciation events. So the pairwise method strongly supports, is strongly consistent with the ortholog conjecture. Our phylogenetic approach on the exact same data strongly rejects the ortholog conjecture. We thought, whoa, okay, that's a little bit stronger result than we expected. So then what we did is we simulated the evolution of expression on these exact same gene trees under these alternative hypotheses. So first we looked under the ortholog conjecture, and that's the pairwise plot that we get. So the OC is indicating that the plot is consistent with the ortholog conjecture. So that's good. We get the pairwise results showing support for the ortholog conjecture. And we get a phylogenetic approach showing result for the ortholog conjecture. So when we simulate the data under that model, we recover that result with the method. If we simulate the data under the null model, that there's no difference, we get a distribution that looks just like our distribution up there. So if you note, all three of these pairwise plots look very similar, and are all consistent with what the expected prediction of the ortholog conjecture was. But the phylogenetic plots are different. They're giving different results when we simulate data under the ortholog conjecture versus the null hypothesis. So not only are the results different between the pairwise and the phylogenetic methods, the pairwise methods don't vary depending on what process you're actually describing the pattern of their invariant. So the phylogenetic methods provide distinct testable predictions, whereas the pairwise methods are not. So what is this pattern that we're seeing here in this tree, if it's showing up in every single analysis? Well, as expected, because we're looking at tau here, that this pattern was telling us something about trait evolution, the evolution of expression on the tree. But that's different in those different scenarios. But what's constant? Well, the tree is constant. And we know from phylogenetic least squares, from all of these comparative methods, that the tree describes the covariance structure that's expected for the evolution of your trait. And this is a plot of covariance. That is just an alternative representation of the structure of the tree, not a description of the evolution of the trait on the tree. And if you'd like to talk to me after the talk, I can explain exactly how that emerges. So this was a very striking pattern. It was, when we went into this, we didn't know how different the phylogenetic and pairwise methods were going to be, but they were even much more different than we expected. We've been in really close touch with Mark Robinson-Rachavi, the author on this paper on the left, who's been incredible in providing feedback on our manuscript, providing code and a variety of conversations as we've gone through these analyses. If you go back in the literature, he's actually proposed how important it is to use phylogenetic approaches to look at these data. So he knew phylogeny was important. We thought it might be, but it really surprised all of us how important it turned out to be in this case. All right, so it turns out, let's revise that statement I put up earlier. So understanding how the evolution of gene function differs between orthologs and paralogs does not explain much about the association between genes and phenotypes. Elicited in this clade when the phenotype that you're looking at is gene expression. So our results indicate that phylogenetic relationships are very useful for understanding the gene function, and the information about orthology and pairology is just providing little additional information. All right? So if you want to describe big patterns in functional attributes of genes, you should figure out like what things explain the most of that. And so essentially, you know, for example, we should expect closely related paralogs to be more similar in function than distantly related orthologs. It's just that sometimes we tend to think of orthologs as being more closely related, but it's not the orthology that's making them more similar, it's just the tendency sometimes for them to be more closely related in the tree. So if that's the case, why use orthology and pairology as proxies? Just talk about how distantly related things are in the tree. It's a much more direct perspective on the processes at hand. So the other issue is that pairology and orthology can have really clear interpretations of small, simple gene trees, all right? But most of us don't have small, simple gene trees anymore. So we have these big gene trees, hundreds of tips often, all sorts of duplication and speciation events in there. When you try and talk about whether two things are orthologs and pairologs, it starts to break down often. So the fact that the terminology breaks down and gets really cumbersome to use, it should, you know, be an indication that the utility of the concepts themselves are really breaking down. So I would say orthology and pairology are unnecessarily complicated and in fact quite overrated. So as it gets trickier and trickier to apply these terms, what are we doing? We're coming up with more of these terms: in paralog, out paralog, within species paralog, between species paralog, all these different terms. It's like Ptolemaic, you know, adding glass spheres to get the orbits of the planets to fit with the observations. It's time to change the model, right? So I think we should get away from these terms. So to analyze past evolution processes, we should look within the trees and not at the tips, I think. So let's think about what orthology and pairology are. So orthology and pairology is a statement about events that happen deep in the tree, right? The duplication and speciation events. Then what you do is, based on those internal duplication and speciation events, you assign genes as being orthologs or paralogs to each other. But really, we want to ask mechanistic questions about things deep in the tree. So we often know if they're ortholog, what the duplication and speciation history is. We tell someone whether they're orthologs or paralogs. Then they have to back it back out to whether there's been duplication or speciation events. So let's just talk about duplication and speciation and forget about moving the annotations to the tips of the trees and then back into the deep tree. So just as an exercise, whenever you're about to use the words orthology and pairology, I suggest that you instead try to explain your idea in terms of speciation and duplication, and I think you'll just feel a little weightlifted, get a little bounce in your step. So there are times to get comparative, there's still time to get comparative functional genomics in a solid phylogenetic footing before it really takes off. And I think this is really important because it's going to get harder to do later. We could gain more insight now. But it's not going to be very effective if we just make appeals to authority and say, well, we all know phylogeny is really important. You should do it. I think it's really important that we make the impacts of phylogenetic comparative methods really concrete for other fields that are doing comparative things, and do as much as we can to implement and build the tools and methods that they'll need for this data. So the concrete examples, I think, are going to be really, really important moving forward. To provide these usable tools. So what are the ultimate limitations of phylogenetic comparative functional genomics? So this integration that Stacey talked about bringing molecular genetics and population genetics into a phylogenetic comparative context. How many of you have heard this when you talk about comparative functional genomics, right? Yeah, so let's see hands. Yeah. How many of those hands are from NSF panels? Okay. All right, so the setup here is that it's two opposite extremes. You can be descriptive over here, or you can be hypothesis-driven. All right, and that's the idea that they're opposite ends of an axis, and we talked about this with Captain Monroe and I in this review that's linked down over here. I would propose that these are not opposite ends of one spectrum, but instead orthogonal axes. So the way we do science is actually that we have a hypothesis and we make a prediction, right? And then we test that prediction with new data, all right? Nothing there says those new data have to be experimental. They can be descriptive. So raise your hand if you believe that the Earth goes around the sun. All right?
No experiment.
No experiment. No one has CRISPR'd the Earth, you know, to check and see if it goes around the sun or anything like that. There is no experiment. Astronomers had this idea and made predictions that they could test with new data that they then collected. So work can be hypothesis-driven and experimental. That's a great way to isolate variables in some systems. Or it can be descriptive, which is a great way to get at processes that happen at scales of time and space that are far larger than we ourselves are. And you can use experiments to do hypothesis-seeking work. That's what Galvani did when he just randomly hooked frogs up, you know, to electrodes and saw them twitch. Or you can do hypothesis-seeking work with descriptive work where you're in a totally new system and you don't know which genes are involved at all and you're looking around. So we could cross that out. I think comparative functional genomics are descriptive and can certainly be hypothesis-driven. The other thing that you hear a lot is there won't be enough statistical power to associate functional genomics with changes in other phenotypes of interest. This is just a number of observations versus variables that you're trying to disentangle. That's a totally legitimate criticism. Something I really encourage people to do simulations before they get into these analyses, figure out how much data they're going to need. And we're going to need to embrace homoplasy. So as systematics, as a community, is so homoplasia averse because it messes up our taxonomy and that's sort of the heritage of the field. So we tend to get uncomfortable when there's lots of homoplasia. Homoplasia is great because it's multiple independent changes. It's statistical power. Improve taxon sampling, as Stacey said, and combine these approaches with other types of evidence. It's really exciting to use comparative functional genomics to get down to smaller gene sets and then go in and use CRISPR or a variety of methods in a very targeted and much more effective way. On another note, so next week, after 10 absolutely wonderful years at Brown, my lab is moving to Yale. So if you're interested in any of these things, please contact me. The lab will be growing after we get there. And again, there's links to notes there. Okay, thank you. Are there any questions for either of us?
Yeah, we have a couple of minutes for questions. I think if you just talk loudly, probably.